Welcome viewers. Welcome to the second lecture on bacterial conjugation. In our first lecture on bacterial conjugation, we have discussed the experiments performed by Lederberg and Tatum and the YouTube experiment by Bernard Davis that helped the establishment of bacterial conjugation. We have also discussed the role of F plasmid or fertility plasmid and how it gets transferred from the donor to a recipient. We highlighted on the involvement of TRA genes, specifically the DTR component and the MPF locus, and the molecular mechanism of the entire process has been discussed. Today we will start our talk with mobilizable plasmids. As we have discussed that mobilizable plasmids are those that need the help of self-transmissible plasmids for their transmission from a donor bacteria to a recipient bacteria. So let's precisely focus that what is a basic difference between a self-transmissible plasmid and a mobilizable plasmid. Basically, self-transmissible plasmid comprises of the MPF locus and the DTR components. On the other hand, mobilizable plasmid contains the DTR components only. It is devoid of MPF locus. So, the mating bridge formation genes are absent in a mobilizable plasmid. But it contains the cis components, specifically the ORI-T site is present within an mobilizable plasmid, which is being recognized by the other components coupling proteins of the self-transmissible plasmid, and thereby they help in the transfer of the mobilizable plasmid. Mobilizable plasmids are really very much important from the viewpoint of their use in recombinant DNA technology. Why? Because their sizes are very small as they don't contain the 15 genes responsible for MPF locus formation. So, if we have any plasmid wherein we have introduced any gene of our interest, and if we want to transmit that plasmid into a desired host, then what we can do? If we introduce the component of a mobilizable plasmid into such a plasmid where our gene of interest has been introduced, then a self-transmissible plasmid present inside the bacterial cell will be responsible for its transfer into a desired host. And if we use some promiscuous plasmids having a broad host range, then the gene of our interest bearing plasmid, which has been converted into a mobilizable plasmid, can be transferred into a broad host range. So thereby, the mobilizable plasmids have really attracted the scientists.
But ma'am, what will happen if self-transmissible plasmid and mobilizable plasmid both belong from the same incompatible group?
Yes, there we need to take an intelligent decision. As we all know that if two plasmids belong to the same incompatibility group, then they cannot coexist. So it's really a difficult job for the coexistence of a self-transmissible plasmid and a mobilizable plasmid belonging to the same ink group. Under these circumstances, they prefer to opt a methodology which is called as triparental matting. Herein what we do, we take 3 different bacterial strains. One bacterial strain will contain the self-transmissible plasmid, which we call as parent 1. And the second bacterial cell will bear the mobilizable plasmid called parent 2. And the third one is called as the recipient bacteria. And if these 3 cells are fused together or allowed to be in contact, then what will happen? In the first stage, the self-transmissible plasmid will be transferred from the parent 1 to the parent 2. And as because self-transmissible plasmid entering the newly recipient cell will start expressing the MPF locus, pilus formation genes, coupling proteins, and finally they will bind to the ORI-T site of the mobilizable plasmid, and they will carry on the mobilization or transfer of the plasmid from parent 2 to parent 3, which is the real recipient. So triparental mating is supposed to be a convenient way to carry on the transfer of a mobilizable plasmid, taking help of a self-transmissible plasmid which also belongs to the same incompatibility group. Usually during bacterial conjugation, we have seen that it is the plasmid DNA that is getting transferred from the donor bacteria to the recipient bacteria. But there are cases where we have seen that the plasmid, while it's transferred to the recipient bacteria, sometimes carry bacterial genes from the donor bacteria to the recipient bacteria. So obviously the question arises that how the plasmid is carrying bacterial chromosomal DNA along with it. So the simple answer lies that the plasmid has got the tendency to integrate inside the bacterial genome. So where it will integrate? So actually the plasmid, especially the F plasmid, contains insertion sequences or IS elements are present within the plasmid genome. And similar IS elements are also present within the bacterial genome. These are the sites which are responsible for the integration of the F plasmid inside the bacterial chromosome in a donor bacteria. So such a bacteria wherein the F plasmid has integrated inside the bacterial chromosome is called as an HFR bacteria, where HFR stands for high frequency of recombination. So why we call it as a high frequency of recombination? As because whenever such an HFR strain tries to participate in a conjugation event with an F minus bacteria, means a bacteria devoid of F plasmid, then what happens? That the transfer starts at the ORIT site, and to transfer the entire bacterial genome, it actually takes 100 minutes. And as we know that 100 minutes is a long time, and for such a long time the mating pair bridge won't exist, as because of existence of strong regulation in the synthesis of the tra-operon genes. So what will happen? The net result will be the half transfer of the F plasmid and partial transfer of bacterial gene inside the recipient bacteria. So the DNA which is received during the conjugation process between an HFR and an F minus mating inside the recipient is a linear DNA. So what is the fate of such a linear DNA which cannot circularize? As it cannot circularize, it cannot replicate itself and it won't be transmitted to the daughter cells. On the other hand, as it is remaining as a linear DNA, it will be disrupted by the nucleases present in the bacterial cell. So the only option that is left with this linear DNA is to undergo some sort of gene exchange with the similar bacterial genes which are present within the genome of the recipient bacteria. So the net result will be sometimes very beneficial to the recipient. Say for example, if I take the HFR bacteria, leucine plus or methionine plus. On the other hand, the recipient bacteria was leucine minus and methionine minus. But after the conjugation event between HFR and the F minus cell, we will see that the recipient, which was auxotroph with respect to these amino acids, will become prototroph about this amino acids. So this is the benefit that a recipient bacteria gets out of an HFR cross F minus mating. So the net result of HFR cross F mating is that the recipient still remains F minus, because the F plasmid is never transferred completely if it gets integrated within the bacterial genome. HFRs are really very important for carrying on the bacterial mapping, as we will discuss just now. But as we know that the plasmid has integrated inside the bacterial genome, leading to the formation of HFR, then there are chances of excision of the plasmid as well. What will happen if the plasmid excises off? If the excision is perfect, then it is the old story back that the cell becomes F plus again. On the other hand, if the excision is improper, then there is some twist in the story. It means that the plasmid, if gets excised off improperly, then it will take some part of the bacterial genome along with it during the excision process. And such improperly excised plasmid is called as a F prime plasmid. So what is an F prime plasmid? It is F plasmid plus containing some genes of the bacterial origin wherefrom it has excised improperly. So what is the fate of an F prime plasmid? Once this F prime plasmid is synthesized, if it participates again in a conjugation event with an F minus bacteria, then the benefit here that we get is that the F plasmid can be completely transferred onto the recipient bacteria, as because its size is quite small, which is sufficient for the transfer of the F prime plasmid. So what is the fate of the recipient bacteria that receives the F prime plasmid? As because now it has become F plus because of the presence of the plasmid, in addition it has procured certain bacterial genes in gift or in excess. So corresponding to those genes which are already present in the recipient bacteria, they become merodiploid or partial diploid. It means that the recipient bacteria is bearing 2 copies of few of the genes that it has procured via the F prime cross F minus matings. So we can consider that F prime cross F minus mating is leading to the formation of a F plus cell. In addition, it becomes merodiploid with respect to few of the genes. And moreover, as the F prime plasmid is a complete F plasmid, it can replicate inside the recipient bacteria and it will be transmitted to the daughter cells. So HFR strains are very much useful as they were used for the first time for mapping of the E. coli genome. So this concept was first established by 2 eminent scientists, Francois Jacob and Elie Woolman, in the year 1950. Where they started their work with an HFR strain which has got a different genotype and tried to mate it with a recipient bacteria with alleles which are different. Means the alleles of the donor and the recipient were different. Say for example, here in the example we will see that the donor was leucine plus azide resistant. It means it was resistant towards the toxicity created by azide. Then it is ton R. It means it is resistant towards infection with T1 bacteriophage. It is lac plus. It means it has got its lac operon on. Gal plus, it was streptomycin sensitive. And this was being mated with an F minus bacteria that was basically auxotroph with respect to threonine and leucine, but it was sensitive toward azide and it was sensitive towards infection with T1 bacteriophage, and it doesn't have lac operon and it is not having gal operon as well. On the other hand, it was resistant towards streptomycin. When these 2 cells were fused and the time for their mating was interrupted at different time intervals, means a kinetic study was done. The cells were allowed to have a contact for a definite time. After that, the mating bridge was being broken with the help of a kitchen blender. And in this way, they could establish the sequence in which the genes are getting transferred. And one more important thing, that to avoid that the donor bacteria is viable, they plated the mixture of bacteria in a media containing streptomycin. And as I said, the donor was streptomycin sensitive, so under no circumstances donor which is a prototroph will survive. So if we get any colonies in the streptomycin plate and which is a minimal media containing plate, then it is obviously a conjugated strain, as because the recipient bacteria is auxotroph. So it is unable to grow in minimal media and donor is unable to grow in the media as because streptomycin is added. So if we get any colonies in the plate which is of minimal media and which contains streptomycin, then it is obviously a result of conjugation between an HFR and a F- cell. In this interrupted experiments, they could establish the sequence in which the genes are getting transferred, and it not only helped us to get the gene order, but we can also see that the frequency of recombinance that we are getting is gradually decreasing as the distance of the gene increases. So, the time which is needed for the transfer of distal markers will be much higher, and as a result of which it will be affecting in the number of recombinants that we are getting. So, herein we can see that how interrupted mating experiments helped the scientist to establish the gene order, and by performing these type of mating experiments, for the first time scientists were able to establish that the E. coli genome is circular, or the circular map of E. coli was established by HFR cross F- mating experiments. Herein you can see that after say 8 minutes, this threonine and leucine genes are getting transmitted together. It means these two genes are so close that it becomes difficult for us to distinguish their distances. Then we could see that azide gene is followed by threonine and leucine, then ton gene is after that. Then we have seen the existence of lac gene and gal gene. So this is how we can carry on the mapping in a very nice way. So these experiments have really helped the scientist to carry on the gene mapping, which is a real benefit of studying the conjugation mechanisms. So here you can see that the benefit of HFR cross F- mating is very much prevalent, as because within the bacterial genome, as I said, there are many insertion sequence elements. So there are many places wherein the F plasmid can integrate. So if you start your story with different HFRs, wherein say for example it has integrated in IS1, and the other F plasmid where it has integrated in the IS2 site, and the third one where it has integrated in the IS3 site, and you carry on interrupted experiments with 3 different HFRs, then you will get 3 different maps. And if you align these maps together, then you will get the entire mapping of the bacterial genome. As I said, by one type of HFR cross F- matings, you won't get the entire map. The reason behind is that entire transfer takes 100 minutes, as I discussed just now. So to get the entire map, you need to work with different HFRs. It means the different regions wherein the F plasmid got integrated.
Ma'am, so far we have learnt about bacterial conjugation in gram-negative bacteria. So are the mechanisms the same in gram-positive bacteria as well?
The case in gram-positive bacteria is very interesting, as because in case of gram-negative bacteria, we have seen whatever changes are occurring or whatever events are being executed, all molecular mechanisms are occurring inside the donor bacteria. We have really don't seen any involvement of the recipient bacteria other than receiving the DNA. But herein, for the first time in case of gram-positive bacteria, you will see that the recipient bacteria is the one that is giving the signals, and that signals are being received by the donor bacteria, and that signal is transmitted inside the donor bacterial cell, which switches on a class of tra-operon genes which are responsible for the mating pair formation or the bridge formation, and the transfer apparatus is switched on. So it depends on the signals which are secreted by the recipient bacteria. So obviously question arises in your mind that what is the nature of the signaling peptides. So basically this signaling peptides are a part of large lipoproteins which are synthesized inside the bacterial cell. These are basically the signaling molecules. Right? The signaling molecules are basically needed for the transfer of the protein onto the sites where they are going to function from the site where they are synthesized. So during their movement, what happens that the signal peptides are chopped up. And we have seen that during its movement through the membranes, from the C-terminal end, say 6 amino acids are degraded, and then the functional signaling molecule is synthesized and it is secreted out of the recipient bacteria. And it gets accumulated inside the bacterial cell, and when it reaches a threshold concentration, then only this signaling molecules will bind onto the receptor proteins which are present onto the cell surface of the donor bacteria. So as we can talk here, take the example that the recipient bacteria is secreting a signal, we are calling it as CAD1. right? The naming of the signaling molecule is directly correlated with the nature of the plasmid which it is going to attract. Say for example, here the name of the plasmid is PAD1, which is being transferred from the donor to the recipient. So the signaling molecule that invites the entry of the plasmid PAD1 is called as CAD1. So it comes here, and what happens next, that you can see the existence of this molecules TRA-B and TRA-C onto the cell surface in the donor bacteria. Once these signaling molecules are coming up, they will bind to this TRA-C protein, which is onto the cell membrane of the donor bacteria. Then it is internalized, and what it does, it binds to TRA-A. TRA-A is basically responsible for repressing the expression of TRA-operon genes. Once this signaling molecule binds to TRA-A, it will remove this TRA-A molecule by changing its conformation, and as because the repression is lifted up, the all class of TRA-operon genes are switched on, which are responsible for synthesis of many proteins which adhere onto the cell surface of the donor bacteria, and the aggregation of these proteins is responsible for making contacts with the recipient bacteria. And this leads to basically the transfer of this entire PAD1 plasmid from the donor bacteria to the recipient bacteria. But there is another interesting observation, that after the event of conjugation, the recipient bacteria now has received the plasmid, then it no longer wants to receive the plasmid again because of questions of incompatibility. It means it has to do something so that the second transfer of such plasmid is restricted, as because the signals are generated constitutively which are responsible for igniting the transfer of plasmid from donor to the recipient. So in this regard, we have seen that the plasmid which is getting entry inside the bacterial cell codes for another smaller peptide which we call as IAD1, means I stands for inhibit. It is similar to CAD1, which was acting as a signaling molecule to initiate the process of transfer from donor to recipient. This IAD1 has got higher affinity towards the TRA-C protein, which is now generated onto the cell surface of the newly becoming recipient bacteria that has now become equivalent to a donor now. So what will happen, that it will block the binding of the signaling molecules, thereby it restricts the entry of similar plasmids again inside the bacterial cell. But there lies another interesting thing, that if the bacteria wishes to procure plasmid of some other type, right, then it is free to do so, as because the signaling molecules for different plasmids are different. So herein, in case of gram-positive bacteria, we can see for the first time that the recipients are actively participate to carry on the process of conjugation.